these are farm pests, and what they do is transmit disease to plants like squash or pumpkin or zucchini, any plants in that cucurbit family. What I'm trying to look at is the symbiotic bacteria that's inside them. So this symbiont that's inside of these bugs can give these bugs fitness advantages. So they are more likely to reach adulthood quicker, and they just survive better. Like we found previously in our lab that with this certain bacteria, this Burkholderia, they needed to survive to adulthood. Otherwise, they kind of fail after their third instar stage. They don't really make it to adulthood. So my experiment is just looking at when, like at what developmental stage they acquire this symbiont, and if it's maybe like a vertical transmission, like from the parents, or a horizontal transmission from the environment. And so I had them in ethanol, and I'm basically leaving them in this Carlson solution, which is just a bunch of salt, and it'll help rinse off some of that ethanol, because the bacteria that's inside of these bug guts, you know, they would probably die if I had any ethanol on the surface still, so that's why I'm rinsing them here in this Carlson's. So I'll leave them there, and I'll get my dissection station ready. So the first thing I'm going to do is lay down some Carlson's solution, and this liquid is what I'm going to use to dissect my squash bug in. Just really easy to get all the guts and everything when it's a wet dissection. So I'm going to lay down a main puddle, and it doesn't matter how much there is, just enough to fit your squash bug. And since this is an adult, I need a little bit more. And I'm going to have like a secondary puddle, so when I cut off extra appendages like his legs, I have an extra area to put them in. And lay him down. Okay. This adult is a male, and you can tell that by this penis here. For females, there's a slit. So the first thing I'm going to do is cut off his legs. And then I'm going to put these legs in that secondary puddle that I laid down earlier. And just keep going.
You're like a surgeon.
I like to pretend that. Okay, so yeah, so the first step I'm going to do is make this first cut right here along the edge. Okay, so that's done. The second cut I'm going to do along this midsection right here, right under his legs. So squash bugs can also give off a really stinky scent if they feel threatened. And the gland is here. I think it's this structure here where there's a hole, but basically you know that there's a squash bug around because their scent is really distinct. And I've been telling people this, but if you ever eat a green Skittle, that is what the squash bug smells like. That taste of the green Skittle, that's the squash bug. It's really weird, but it's true. Okay, almost there. Okay, so now my cuts are finished. I'm going to switch my tool from the scissors to another set of forceps. And now I'm going to try and open him kind of like a book. And we'll be able to see his midgut. Alright, so there's a lot of connective tissue that I will have to try and scrape off of his exoskeleton. To try and lift off this abdomen, this exoskeleton right here. And like, at least with my experiment, that's all I really need to do. Okay, so now you're getting to see the midgut structure of the squash bug. These red things are his testes, and these are different parts of his midgut. The important one that I am looking at is this
white organ that's a little curly and grooved. That is the M4 crypt, and that is where the symbiotic bacteria will be. So with a lot of these like experiments that we do, mostly we isolate just this part, but for me, I'm still like beginner, so I just take the whole thing. So what I did with my experiment, I fed these bugs at a young age. I fed them fluorescent burkholderia, and I'm tracking this fluorescent burkholderia. So when I take out these guts, I'm going to crush them and I'm going to plate them on LB plates, and I'm going to see if there's any fluorescent bacteria colonies that grow. So that's why for me it's not really important to like concentrate on just isolating this M4 crypt. I can just take out the whole thing, because as long as there's fluorescence, that means it works. I'm just going to extract all of this. Let's take it all out. So this is the squash bug net gut, and with that my dissection is complete. I'm going to put this in more Carlson solution so I can keep it until I crush it and plate.
We have a male monarch butterfly, and you can tell the sex difference between male and female, because males, they have these two little claspers down here to grab and take a hold of the female during mating. So this is a monarch butterfly, and we work with this in our lab. However, butterflies, they don't come from their mothers looking straight like this. They have to go through metamorphosis, which is very distinct developmental stages, to become this beautiful black and orange butterfly. They lay these little tiny yellow dots on a milkweed plant. So it's funny because these huge butterflies come from these small little yellow dots. And in this box we probably have about 60 or 70 eggs. But what happens is the caterpillars, they hatch from these eggs and they grow. So cool fact, caterpillars, they grow 1,000 times their original body weight in order to become a pupa. So although I don't have a big caterpillar here today, I have what we call first instar caterpillars. So this is from the egg I just showed you, from when the caterpillars hatch out the egg. So they become first instars, and it's pretty much, instar means molt, actually, like grow out of their head. So they extend from the head every time they molt. So this is the first instar. In about 2 or 3 days, it'll become a second instar where it'll shed its head lining and it'll grow forward and become bigger. So one question we're interested in answering in our lab is based on the different plants that these caterpillars feed on. Do these plants change or affect the bacteria living in the gut of these caterpillars? So today I'll just be feeding these newly hatched caterpillars on one of their host plants. And this is a particular species that has a high nutrients and a low amount of toxins that plants produce. Along with our question and being interested in how different plant species affect the bacteria that live in the gut of the caterpillar, we have to control for taking the caterpillar off the plant when they first hatch. So in nature, there will be eggs naturally under the bottom of this leaf side. I should do it like this, excuse me. Under the bottom of this leaf side, and those caterpillars, they then hatch, they eat their eggshell, and they crawl around until this whole plant is gone. But for the purposes of our experiments in lab, we take little caterpillars and we add them to the diet of our choice. So I'm just taking these tweezers and I add them on, and these caterpillars begin to eat, and I then take them off and try to figure out what bacteria are in their gut. Caterpillars, they also make silk. Cool fact, so spiders aren't the only insects that make silk. And the point of the caterpillars doing this, that in nature, if the wind does blow, since they're so dependent on the plant they eat on, they need a way to hold on to the plant if they were to get knocked off. So it's like a stress mechanism. They know to spin silk from themselves to hang on to the plant.